Have you ever really thought about what happens inside a tumor? I mean, down at the cellular level. Wondered what makes some cancer cells just take off, growing faster, spreading more aggressively than others right next to them. For years, that's been, well, a really tough puzzle for scientists. But today, we're doing a deep dive into something pretty revolutionary. It's a computational algorithm called Sprinter, and it's starting to give us this unprecedented clarity into how tumors proliferate and, crucially, how they metastasize. Our mission today. To unpack how Sprinter actually lets scientists see this growth, this spread, with frankly astonishing clarity, we'll look at the surprising things it's already revealing about basic cancer biology, and maybe most importantly for you, what this could all mean for the future of treating cancer. And just so you know, our source material for this comes straight from a cutting-edge scientific study deep in the world of single-cell genomics. So we're getting this info hot off the press, so to speak. Okay, so let's set the stage a bit. Understanding how cancer cells multiply and spread, it sounds basic, but it's always been a massive challenge. Researchers really struggle to pinpoint individual cancer cells right when they're most active. During the S phase, when they're copying all the DNA, or the G2 phase just before they split. These are like the busiest moments for a cell, super dynamic, really hard to track accurately, and even if you could catch them in the act, trying to figure out which specific genetic clone they belong to within that jumbled mix of cells in a tumor, that was just a huge technical barrier.
You've absolutely nailed the core issue there. That difficulty created a really significant blind spot in single-cell cancer genomics. Before this, studies often had to rely on, well, less precise methods, bulk measurements, things like Ki-67 staining.
Right, which tells you something is growing, but not who exactly.
Exactly. It gives you a general average, not the specifics. Or they'd use single-cell RNA sequencing, looking at gene activity. But that doesn't directly tell you about the DNA duplication process itself or the cell cycle phase in the same way. The fundamental problem was these methods just didn't have the resolution. They couldn't really profile proliferation clone by clone, and they couldn't link that proliferation directly to how the clones were evolving, using the DNA sequence data itself. Left a lot of big questions hanging.
Okay, so this is exactly where Sprinter comes in. It's positioned as this novel solution, right? Think of it like a really smart computational tool, designed specifically to slice through those old challenges. It's built to figure out clone-specific growth rates using data from single-cell whole genome DNA sequencing. scDNA-seq, they call it. It's like switching from a standard magnifying glass to a super high-powered microscope for looking at tumor growth.
And one of the really clever bits, the innovation that makes it work so well, is what they call its replication-aware correction framework.
Replication-aware. Okay, what does that mean in practice?
Imagine trying to get a sharp photo of a race car speeding past. It's likely to be blurry, right? S-phase cells are like that their DNA is actively being copied. It's dynamic. It's messy. This framework acts like an advanced image stabilization system. It allows Sprinter to account for that ongoing replication. And because it can do that, it lets us accurately see changes in the DNA structure, even in these busy S-phase cells. Things like DNA copy number alterations, or CNAs.
Ah, CNAs, where cells have too many or too few copies of certain DNA sections?
Precisely. Gains or losses of big chunks of the genetic instruction manual. These often drive cancer growth, but seeing them clearly during replication, that was the bottleneck Sprinter overcomes.
Okay, so they built Sprinter, they tested it, then came the moment of truth. Applying it to actual cancer cells, what did they find? They used it on a really big data set, didn't they? Almost 15,000 non-small cell lung cancer cells. NSCLC.
That's right. And crucially, it was longitudinal meeting samples over time from both the main tumor and from metastases, the spread out sites.
And they didn't stop there.
No, they also ran it on public data. Triple-negative breast cancer, high-grade serous ovarian cancer, just to show it wasn't a one-off, that the tool could work across different types of cancer.
Which is huge for showing its potential usefulness,
Definitely. And the first major finding that jumped out was this. Clonal heterogeneity and proliferation is, well, it's everywhere. It's widespread. Meaning a single tumor isn't one uniform mass of cells all behaving the same way. It's more like a bustling city with different neighborhoods, different groups of cells. These clones growing at vastly different speeds. Some clones are dividing really rapidly, others are much slower, almost dormant. It's a really mixed bag.
Okay, and here's where it gets really interesting, right? Linking this growth speed to how cancer actually spreads.
Exactly. This is, I think, one of the most impactful findings. Sprinter showed that the clones that are dividing the fastest, the highly proliferative ones, those are the ones much more likely to break away and seed metastases. to start new tumors elsewhere in the body.
Wow. So it's the speed demons driving the spread.
That's what the data strongly suggests. It establishes this direct link at the clonal level between how fast a cell is dividing and the tumor's potential to metastasize. That's a connection researchers have been trying to pin down for a long time.
That really clarifies a key part of the process.
And there's another practical link here, something relevant for patient monitoring. These fast-growing clones, they also seem to shed more DNA into the bloodstream, what we call circulating tumor DNA, or ctDNA.
Ah, the basis for liquid biopsies?
Precisely. So if you do a blood test, a liquid biopsy, you might be preferentially picking up signals from these more aggressive, highly proliferative clones. This finding really bolsters the value of ctDNA monitoring. It suggests it could give us clues about the most dangerous cells within the tumor, potentially helping guide treatment choices, like a non-invasive surveillance system.
Okay, so we know some clones grow faster, and those are the ones more likely to spread and show up in ctDNA. The big question then becomes why? What's making these specific clones proliferate so aggressively? Is it all down to their genes?
That's the next logical step, yeah. And Sprinter helped dig into that too. The answer seems to be, well, it's complicated. It's not just genetics. Sprinter found correlations with both specific genetic changes, things like known cancer-driving oncogene amplifications. KRAS amplification was one example.
Okay, so specific mutations or gene copies do play a role.
Yes, definitely. But also, interestingly, with non-genetic factors. Specifically, something called altered replication timing, RT.
Altered replication timing. Okay, break that down a bit.
Sure. So think of the genome, all the DNA, as this enormous library that needs to be copied perfectly every time a cell divides. Replication timing is the precise schedule, which books get copied when, in what order, at what speed. Altered replication timing, or RT, means that schedule gets messed up. Certain parts of the genome start getting copied earlier or later than they're supposed to.
So it's not a change in the DNA sequence itself, but in the process of copying it.
Exactly. It's a change in the dynamics of DNA replication. And the study found that these alterations in timing also correlate with how fast the clones are proliferating.
That's fascinating. So it's this mix of genetic mutations and these functional changes in cell processes driving the growth.
Right. It gives us a much more complete picture, doesn't it? Moving beyond just the static DNA code to include the dynamics of how the cell actually works and divides, it opens up new ways to think about what fuels tumor growth.
Definitely a more nuanced view. Okay, let's shift gears slightly and evaluate the study itself. Sprinter sounds powerful. What are its main strengths?
Well, the big one is just the novelty of the method itself. It really does fill a significant gap in how we study cancer at the single-cell level. That's a major contribution.
And they validated it, made sure it was accurate.
Yes, they did robust validation. Tested it against a known ground truth dataset cells where they knew the ploidy of the DNA content to show it could correctly identify cell cycle phases. And critically, they showed it outperformed older methods, algorithms like CCC and MAPD, which were trying to do similar things but lacked Sprinter's precision, especially with DNA copy number changes during replication.
Plus, as we mentioned, its ability to work across different cancer types, lung, breast, ovarian, that scalability is a real plus.
For sure. But like any study, especially one introducing a new method, there are limitations to keep in mind. It's important to have that balanced perspective.
Okay, what are some of those?
Well, for one, detecting cells very early in S phase is still a bit tricky for the algorithm. Its sensitivity isn't quite as high in that specific narrow window.
Right.
And identifying cells in the G2 phase just before division is maybe less robust because it relies mainly on just the total amount of DNA sequence reads, not quite as sophisticated as the S phase detection.
Okay, understandable limitations for a new tool. Anything else?
A pretty significant point is that their strongest conclusions about tumor evolution, how clones change over time and spread, are based mostly on that one detailed, non-small cell lung cancer patient dataset.
So while the tool works broadly, the specific evolutionary story might need more confirmation in other patients and cancers.
Exactly. It's a fantastic deep data set from that one patient, but you always need broader validation before you can say those specific evolutionary patterns are universal. Generalizability is key.
Makes sense.
Also, the type of DNA sequencing they used, low-coverage sdDNA-seq, is great for CNAs. But it means they couldn't really zoom in on single-letter DNA changes, single nucleotide variants, or SNVs. So potentially rare but important point mutations might have been missed.
Right, a trade-off with the sequencing depth.
And finally, that fascinating link to altered replication timing, the RT analysis. That was supported by looking at gene expression data from bulk tissue samples, not yet single-cell RNA data linked directly to the sprinter analysis. So correlating art precisely within individual cells is still a next step.
Okay, that provides really good context. So, knowing the strengths and these current limitations, what's the outlook? What does Sprinter mean for the future of, say, precision oncology? Where does this research go next?
Oh, the potential is really exciting. There are several clear paths forward. First, refining the algorithm itself, improving that early S-phase detection, making the G2 phase calls more robust, perhaps by bringing in other data types like cell imaging or maybe RNA data from the same cell.
Making the tool even sharper.
Exactly. Then, the big one, applying Sprinter to much larger groups of patients across many more cancer types. That's absolutely crucial to confirm how generalizable these findings really are. Does clonal proliferation predict metastasis consistently? We need more data.
Testing it far and wide.
Yep. And then think about combining Sprinter with other single-cell technologies. Imagine layering its proliferation insights with single-cell RNA-seq to see gene expression in fast versus slow dividers, or ATAC-seq to see how the DNA is packaged differently. That gives you a multi-omics, really comprehensive view.
A much richer picture of each cell.
Yes, definitely. And digging deeper into that altered replication timing aspect.
Yeah.
How does art evolve over time? How does it connect to other epigenetic changes, the way genes are switched on or off? There's a lot more to uncover there.
But the ultimate goal, I guess, for patients, for you listening, is how this translates to the clinic, right?
Absolutely. That's the key. The really exciting prospect is using Sprinter, or tools like it, to potentially predict how a patient might respond to a specific treatment, or, importantly, how resistance might develop. If you can track, clone by clone, which cells are proliferating under therapy and which aren't, you get this incredibly precise readout of what's happening. Are the aggressive clones being killed? Are resistant ones starting to grow out?
So it could guide treatment in a much more personalized way.
That's the hope. Moving towards therapies that are truly targeted not just at the tumor, but at the specific clones within the tumor that pose the biggest threat, hitting cancer where it's most active and dangerous.
So summing it up, Sprinter really gives us this unprecedented high-definition view right into the engine room of cancer growth and spread. It's revealing these critical details at the single-cell level that, honestly, were pretty much invisible before. A new lens on an old foe.
It really is. And maybe a final thought for you to consider. Imagine the possibilities if we really could precisely track and then maybe even target those specific cancer cells most likely to spread or to resist treatment. How might that completely reshape our approach to cancer care, moving away from these sort of broad assaults towards highly targeted cellular level strategies? It really makes you think about what precision medicine could truly become.